Hello and welcome to Biology Interest. Today we're going to be looking at in vitro gene cloning. We've already looked at how in vivo gene cloning can be used and how we can create gene fragments, but now we're going to be looking at in vitro gene cloning using the polymerase chain reaction. So this is a form of reaction where you want to create many DNA fragments, and you would use a polymerase chain reaction for that. And it means you don't have to use a living cell in the same way as you would with in vivo gene cloning. So you can clone any fragment of DNA, such as genes, using PCR, and it takes a short amount of time to fully synthesize millions of clones of DNA fragments. And it doesn't involve living cells, which is another benefit. So let's outline the basic steps of in vitro gene cloning. So first of all, you would take all the strands of DNA that you've collected so far from the DNA fragments, and the DNA fragments you want. You then take primers and place them in the mixture with the DNA fragments, and you also get loads of free nucleotides which you place in with the DNA fragments. Mixing together with a heat stable, that's important, DNA polymerase, which is the enzyme used to bind all these strands together. So you would then heat the mixture to 95 or between 95 and 98 degrees. This is to split each double-stranded DNA into two single-stranded DNA strands. So we now have two single-stranded pieces of DNA. So we then cool the mixture down to 50 to 65 degrees, between 50 and 65 degrees, which enables the template strands you've added into the mixture earlier to hybridize, almost bind using the complementary hydrogen bonds. So they bind with hydrogen bonds with its complementary primer. So that means you now have template DNA with a short section that's double-stranded and the rest single-stranded, because the template strand is attached with hydrogen bonds to the template DNA you just created. And you do that by cooling the temperature down, which allows the hydrogen bonds to form, because the 95 to 98 degrees you had in the first step is too hot for hydrogen bonds, and it breaks the hydrogen bonds, which is why you get the two single-stranded pieces of DNA. So once you've allowed the template strand to bind with hydrogen bonds, you then heat the mixture back up to 72 degrees. And this allows the addition of complementary nucleotides to each strand of the templated DNA. And this allows the length of each templated DNA strand to extend from the respective primers, forming two copies of double-stranded DNA from where you had one copy of double-stranded DNA beforehand. And both these strands are identical to each other and the original piece of DNA you had, so you've cloned the DNA essentially. And the reason you heat it back up to 72 degrees is because that is the optimal temperature for the heat-stable DNA polymerase you're using, essentially. That's the optimal temperature for it to work the best to bind all these DNA nucleotides together. And the reason you use this high temperature and this heat-stable DNA polymerase is because you don't want bonds to form between the DNA strands, essentially. PCR is widely used in biological medical research, and you use it to determine evolutionary relationship between organisms, genetic disorders, many different types of things, and also crime scenes where they would amplify a trace of DNA they find. So overall, an in vitro technique is often simpler, cheaper, and more sensitive than an in vivo technique where you grow it with a cell. However, in vitro conditions aren't really like a real condition. So if you're looking for a study that wants to reflect the real conditions of actually being inside a cell, in vivo studies tend to be better and may produce better results. But otherwise, in vitro is better for pretty much everything else. So I hope this video has been clear. Please like and subscribe. Stay tuned for more videos from Biology Interest. Thank you.